Good evening, everybody, and welcome to tonight's Midsummer Nights Science lecture, the last talk of the series for this summer. My name is Tom Ulrich. I'm the Associate Director for Science Communications here at the Broad Institute. It's my pleasure to welcome all of you here, and also everybody watching us on Facebook Live tonight. These talks, which take place every summer, as you may remember, are meant to explore key advances in genomics and medicine and foster conversation between Broad scientists and the public. Our speaker tonight is Joel Hirschhorn. Joel is an institute member and co-director of our Metabolism Program here at the Broad. He's also a pediatric endocrinologist at Boston Children's Hospital who studies the genetics of human traits such as height and obesity. Joel is going to deliver a special lecture tonight. It's our Eliana Hechter Memorial Lecture, which he will describe for you in a moment. And during this lecture, he will be talking about recent advances in genetics that have led to discovery of hundreds of or thousands of places in our genomes that influence height or obesity, what they tell us about the biology of human height and obesity, and how these discoveries could lead to new treatments for obesity. A couple of housekeeping items before I bring Joel up onto the stage. If you have any questions, please hold them until the Q&A session after his talk. If you're watching us on Facebook Live, please leave any questions that you might have as comments on the video feed, and I'll work them into the Q&A as best I can. And without further ado, Joel Hirschhorn.
Great. Perfect. All right, so first of all, thank you all for attending tonight's lecture. It's really great to have so many people in the audience, and I'm really looking forward to the question and answer session at the end of the talk. In the talk, How Do Genes Control Our Size and Shape? We'll explore how recent dramatic advances in genetics have changed what we know about how our genome influences our height and our weight, our tendency toward obesity, and potentially how those could lead to new treatments. Tonight, I'm particularly privileged and honored to be giving tonight's talk, which is the Eliana Hechter Memorial Lecture. It's funded by the Eliana Hechter Memorial Fund. And this honors the memory of Eliana, who's a gifted MD PhD student and a member of the Broad community, and whom I knew both as a friend and colleague and briefly member of my lab as well. I want to thank her father, Michael Hector, for being with us tonight, and also acknowledge her partner, Jacob Lurie, who was not able to be here tonight. Before I give the talk, I want to give a brief description of Eliana. She graduated magna cum laude actually at age 18 from the University of Washington in 2006 with a degree in mathematics, then went on to become the second youngest Rhodes Scholar in history. She then did a PhD in statistics at Oxford, where is one of the places I met her. I'll tell a story about that in a moment. And then continued her studies in medical school in the Health Sciences Technology program at Harvard and MIT. In addition to all of that science and medical training, Eliana also is a gifted writer. She studied creative writing with the university's honors program in Rome and had a short story, Extremities, that was published posthumously in the quite eminent literary journal Plowshares. And fitting for this lecture series, she believed very passionately in communicating the excitement and the wonder of math and science to people who weren't scientists and mathematicians. And this, along with really what was kind of one of Eliana's defining features, which is her kindness and thoughtfulness, this inspired her to go to help others to really understand these topics. And as one example, as a 16-year-old, apparently she rewrote a professor's notes in order to make them more accessible to those who were less math literate than herself. I'll talk briefly about the Eliana Hechter Memorial Fund and then about my own personal experiences with Eliana. So last year, the fund was used to create the Eliana Hechter Travel Fellowship, which has enabled several scientists to travel to research symposia and present at conferences to advance their research. And I believe Eliana herself, who's a beloved member of the Broad's program in medical and population genetics, would be really pleased to know about all of this wonderful research and the opportunities that the fund is creating. So as I said, I first spent a fair bit of time in the same place with Eliana during her PhD defense. And in Europe, in certain countries, the PhD defense, the faculty who attend, they ask questions and try to sort of figure out how well the person knows their subjects. They're sometimes even called opponents. So it became very quickly clear two things about Eliana. The first was that she was not one for formality. So in the UK or in England, you have to wear a cap and gown, like the gown that you graduate in, during the defense. And as soon as we got behind the closed doors, the first thing that Eliana said was, "Can I take this hat off?" She was really wanting to just get into the science. And the other thing that became quickly clear was that she just had an amazing deep understanding of the issues in her thesis. And in fact, I still occasionally have opportunities to cite her PhD thesis. In fact, I did it in this very room a few months ago when a point came up that Eliana had actually figured out many years ago. So she actually came to the Broad, and it was clear to everybody she had this blazing intellect, fierce curiosity, and a willingness to state, sometimes very forcefully, strong opinions, but importantly, the ability to back them up. So in short, she just fit right in at the Broad Institute. So I was really delighted when she said that she wanted to work in my lab for a period of time, as she was getting ready to start medical school. And she was there a short time, but actually made quite an impact. She wrote software that we still use a version of to this day to try to find genes that cause, for example, severe short stature in children. And in fact, some of the work that I'll talk about, she was a co-author on. In one thing that was also striking was that when she found a problem that she was interested in, she really devoted herself to it passionately. And some of the things that she helped us do, and then there was a patient that we had who had a sort of unusual form of short stature, and she just went to unbelievable lengths, including arranging collaborators in multiple different countries, including languages that she figured out how to get somebody who spoke them to arrange these collaborations. And we were never able to figure it out, but one of my goals that I hope I achieve before I retire is sort of another way of honoring her memory, is to actually figure out the genetic basis for this patient that this project that Eliana was so passionately involved in. So with that introduction, I'd like to begin this year's Eliana Hechter Memorial Talk, How Do Genes Control Our Size and Shape? All right. So people come in many shapes and sizes, and we know that genes play a strong role in determining how tall or short somebody is, or how lean and obese people are. And you might say, well, how do we know that? Well, essentially, it comes down to looking at people who are either more or less genetically similar to each other and seeing, are they more or less similar in their height and in their weight? So here you can see 3 pairs of identical twins, actually all in the sporting world, basketball, tennis, doubles, and badminton. And you can see that there are many things about these twins that are very similar, and that includes actually their height and their weight. If you look, though, at people who don't have identical or nearly identical genomes, so their DNA sequences of twins are almost completely identical, but if you look at sort of a range of people, and these are some of our family's favorite TV shows on top, and then one of the X-Men movies on the bottom, you look, there's a much broader range, right, than you see in the pairs of identical twins, not just in height and weight, but in many other features that you can see, and actually in many other things that you can't see. So for example, if you had a pair of twins, identical twins, and one of them had type 2 diabetes, the likelihood that the other twin would develop type 2 diabetes somewhere in their lifetime is actually about 80% or more. But there's obviously much less of that concordance between just two random people. And so what this tells us is that if there are more genetic differences between people, then you see more variability in things like height and weight. So the genotype, the version of DNA that you're born with, leads to differences that we can see or measure in people. It's called phenotype. So if we want to understand the basis by which differences in our genes influence our height and weight, one of the first things we want to know is, well, how different are people? So how much genetic difference is there that could contribute to things like height and weight? So shown here is 2,000 letters of our genetic code. This comes from chromosome 17. If you then take this region of the genome and you compare two different versions of it. So it could be the version I got from my mother and the version I got from my father, or it could be one of mine and one of anybody's in the audience's. If you compare them on average, what you'll find is that in these 2,000 different letters, and these are the A's, C's, G's, and T's that make up our DNA sequence, in these 2,000 letters, you'd find maybe two places that differ. So we're actually about 1 in 1,000 different from each other, or put it another way, 99.9% identical genetically with each other. So you can ask another question then. If you discover these differences, so you line up two regions of the genome and you say, okay, here's a place where maybe one person has an A and one person has a T, or one person has a C and one person has a G, then you look and you say, let's say we sequence the genomes of everybody in this room. Would we find both of those versions of the DNA sequence, or would one of them be really rare and unique to one of the people? So let's say in my genome I had a C. Would other people in the audience have a C at that same spot? And the answer is mostly yes. So most human genetic variation is actually shared among lots and lots of people on the planet, and in fact is often shared across continents, so that if you find a variant in Europe, you're almost certain to find it in Africa as well, at least for most of the variation that you discover. You can also ask a different question. Where in the genome is this variation, is most of this variation? So it turns out it's spread pretty evenly across the genome. There's some subtleties about places that have a little more, a little less, but it's basically spread pretty evenly across the genome. And each of our genomes is about 3 billion bases long, so 3 billions of those letters, A's, C's, G's, and T's. And you might think, well, the variants that are going to matter are going to be the ones that are in the genes themselves, and particularly in the parts of the genes that code for proteins. This is called the coding region of genes. So that variant, if you have an A or a C, it might lead to a different amino acid getting incorporated into the protein. The protein itself would be different. It turns out that the parts of the genome that code for proteins actually are only about 1% of the genome, about 30 million bases. And so because the variation is spread out evenly, most of the variation that we have in our genomes is actually non-coding variation. It doesn't directly affect the sequence of the amino acids and proteins that we have in our body. So how much difference do these single base changes? Most of the variation that we see is single base changes. A few of them are more complicated variants that say, delete bases or put extra bases in. Most of them are changing single bases. So how much does changing one base make? Well, we think that most of the time, this has no effect at all. So you have an A, you have a C, doesn't matter. Some of the time, though, that single base change will make a difference. What difference that is, and figuring out, okay, if you change this base, what's the consequence for a person? That's one of the major goals of human genetics today, is trying to make those connections between changes in genotype, whether you have an A or a C at a particular spot, and changes in phenotype. Is somebody short? Are they taller, heavier, lighter, more prone to get type 2 diabetes? And it turns out that these single base changes, at least most of the ones that we've been able to find, have very small effects. So if we're talking about changes in height, for example, it might be that they change your height by a fraction of a millimeter. So the differences in height that you see across most of this slide are actually the combined effects of thousands and thousands of these different places in the genome, each one tweaking you up a fraction of a millimeter or tweaking you down a fraction of a millimeter. And depending on how many shorter or taller versions of each gene you have, the sum of that will basically determine what at least the genetic portion of what your height is. Now, sometimes those single base changes can have a big difference. So this is the X-Men again, and one of my favorite actors here is Peter Dinklage. And he has a single base change that causes a condition called achondroplasia. And in this case, that one single base change actually has a very dramatic effect on height. It's actually, on average, about 16 inches of height difference, at least, for having this single base change. But that's not the typical scenario for the genes that influence our height. What's more typically seen is that there are lots of different variants, and some of them might increase your height or decrease your height, or if we're talking about the risk of diseases like type 2 diabetes or asthma or pretty much any other disease you can think of, they sort of tip a balance one way or the other. So in this case, there are genes that on net increase risk of disease, and here are some genes that on net decrease risk of disease. And the balance is tipped a little bit in favor of increasing risk, so the person who has all of these different variants would be a little bit more likely than the average person to get a disease. And you could make the same sort of balance slide for whether you're likely to be taller or shorter or heavier or lighter. I've drawn all of these genes with equal effects, but actually that's not really the case. Some of them have smaller effects and some of them have bigger effects. So you might have a gene here which has a bigger effect, and that might tip the balance a little bit. And in the case of the single base changes that have a huge effect, where they themselves are sufficient to cause a disease, or sufficient to have huge effects, like for achondroplasia, I should say that these can be either common or rare in the population, but as you get to larger effect sizes, the variants that have these bigger effects tend to be rarer. And the reason is that if a variant has a big effect on a gene, it will actually affect the biology in a little bit more of a profound way, and evolution actually takes notice of that. So if you just affect things by a tiny little bit, evolution actually doesn't care, and those variants can become common in the population. But as the effect sizes get bigger and bigger, then what happens is evolution takes notice, and it prevents those variants from becoming common in the population. So they tend to be rarer. So for example, you might not see it in this room. You might have to have multiple auditoriums before you ever found it. And again, the variants that cause these single gene disorders, or where one gene is enough to cause disease, this is called monogenic disorders, those are almost always very rare. And I don't want to leave the impression, I've been talking obviously a lot about genes and how they determine disease risk and height and weight. I don't want to leave the impression that everything is genetic, right? So some things are mostly genetic. So within a particular population, for example, about 80% of the variation in height is due to inherited factors. So if you take people who are born all around the same time and all in the same country, most of the differences in height that you see will be due to genetic factors. Then there are things at the other end here. So for example, being struck by lightning, and hopefully nobody was struck by lightning earlier today. That is largely an environmental thing, whether you happen to be standing in the path of the lightning bolt. Although even there are genes that influence your risk-taking behavior, so you might be more likely to go out golfing in a lightning storm, and so maybe genes would play a role. So for most things, though, many common diseases, including obesity and things like type 2 diabetes, asthma, it's a mix. So there's some genetic predisposition that you're born with, and there are environmental factors as well, like how much food you have available, what you choose to eat, whether you're exposed to smoke, those sorts of things. All right, so that's kind of the brief overview of how variation in genes, in a general sense, influences variation in things like height or weight. There are these common variants that have small effects, and then there's some things which have slightly bigger effects. They tend to be a little bit rare in the population, and most of the variation that we know about is non-coding variation. So if we study this variation and we say, if we figure out what are the variants that influence height or weight or disease risk, what can we learn from that? And there's really two classes of things we can learn. One is that genetics is a way of understanding the underlying biology of disease or of things like height or weight. And that could guide, for example, new treatments of obesity. If we understood the biology well enough, using the clues that genetics can provide, we could say, aha, we now have a rational way of going about new treatments. And that's really the long-term vision of not just what I'm doing, but what many people at the Broad Institute are trying to do. There's also a shorter-term impact, particularly for the variants that have large effects, or that maybe in combination have large effects, and that they can actually help to provide diagnosis or predictive information to patients. So saying, oh, you have this particular variant, we know that you're very likely to go on to get diabetes, for example, or we understand why you have severe short stature. For most of the talk, I'm actually going to talk about the first part of this, which is how can we understand what variants influence height and weight, and how can we use that to understand biology? So this is one of my favorite pictures, because how often do you see a president of the United States next to DNA over here? The one on the right is the DNA. And this was at the launch of an initiative called the Precision Medicine Initiative, now rebranded as All of Us. And the website for that, and it's mostly a large effort to collect DNA and information from about 1 million people in the United States. But the rationale for doing that starts with far too many diseases really have no proven means of prevention or effective treatment. And in order to make a difference in that, we have to understand, get better insights into the biological and other influences on these diseases. So there's a kind of generic way of trying to make advances in biomedical research. And the idea is that you have a disease that you want to understand better. And you use some method of studying it to get a biological insight. Use that biological insight then to get a therapeutic lead, so an idea that, okay, if you could only turn down this protein or change this protein in a certain way, that might be useful, and then you take that back into people and see if it works. So there are lots of different ways of studying human disease, and one of the ways that we use, and what I'm going to tell you about today, is to use human genetics as a way of gaining these important biological insights. So why use genetics? Why, and a lot of people at the Broad do this, and a lot of people around the world. Why do we like using human genetics to understand biology? Well, one is that it's an unbiased approach. So you might come in saying, I think I understand what causes obesity, right? And so I know it's this protein or it's this part of the cell, and so I'm going to study this protein or this part of the cell. And you might be right? And if you're right, that's great. You now have sort of a head start. But there's no way of knowing. There are lots of guesses that you can make about what the biology is. And it might be biology that nobody's ever heard of before. So there's no way of starting on that experiment without getting the clues as to this unknown biology. One great advantage of genetics is it actually allows you to search across the entire genome, including things you know absolutely nothing about, and the genetics points you to the right place to look. The other big advantage of genetics is that it establishes causality, and I'm going to delve into that in the next couple of slides. So let's say you wanted to understand, you came down from Mars. You said, I want to understand what makes people sick enough to get into the hospital. So the way I'm going to study this is I'm going to go and I'm going to study people in the hospital, and then as a control, I'm going to go outside the hospital and study people outside the hospital and look for differences. And if you do that, the biggest difference you will find is the way that people are dressed, right? So in the hospital, everybody's wearing these hospital gowns. You would say, aha, I know the cause of all illness. It's hospital gowns. If we could only get rid of them, right, we would cure all illness. And you can say, well, obviously that's, you know that sort of they put on the gowns afterwards. But sometimes it's not so easy to figure out which correlations are actually causal and which ones are not. And one of the big advantages, so in case you didn't get that difference, here's a cartoon from XKCD, which I love. And as soon as you get this cartoon, you understand the difference between correlation and causation. So, all right, this is enough laughter that I think people get it. All right, so the other big advantage of genetics is that the correlations that you see between genotype and phenotype are always causal. Again, there's some caveats that you have to have done your genetic study properly, but we think we understand how to do it properly now. And the reason is that you're actually born with the genetic variation. It comes before everything else. And it's actually a random process. This goes all the way back to Mendel and his peas. It's a random process, which variant of the ones that your parents have, which one you're born with. So that randomness and the fact that it comes first means that if you find a correlation between which version of a genetic variant you have and what phenotype you have, so whether you're tall or short or whether you have diabetes or not, there's a causal link between those two. So because of those advantages of genetics, there's been a lot of effort to try to find and discover these genetic correlations. So I'm going to use height as an example, as I will through a lot of this talk. And the way that you look for these correlations is something called a genetic association study. So the first step is you get a bunch of people who are willing to participate in a study, and you get their DNA and you analyze it. And you look for places where there are genetic differences, and you could get their whole genome, sequence their whole genome, or you could look at specific places in the genome where you already know that people vary. And the next thing you do if you're interested in height is you measure their height. And this is, height is pretty much a kind of bell-shaped curve here. And then you compare the heights of people who have different genotypes. So if there's a spot right here that some people have a G and some people have an A, you look for people who have either two A's, one from their mother and one from their father, two G's, one from each parent, or maybe they got one A from one parent and they got one G from another parent. So that's shown on the next slide, actually A's and C's on this slide. And so you look through each of the places that people vary genetically. So on the left here is an example of a variant that has no effect on height. So it doesn't matter if you have AA, AC, or CC, people are on average the same height. But on the right is a place where, aha, it actually makes a difference. If you have two Cs, you're much taller than if you have two As. And the effect size that I've drawn here is actually much, much, much bigger than we would see in a typical association study. Just so that you can see it, typically, as I said, you would have sort of, fractions of a millimeter of difference between the CC genotype and the AA genotype. So you go through and you can systematically look through millions of places in the genome where we know people vary and ask for each one of them which ones influence height or obesity or disease risk. So people have done this, us and many other people, a lot of work actually done here at the Broad. And each of the, what's shown here is a catalog, a visual catalog of the results as of actually earlier this year. And each of the vertical lines here is one of the chromosomes in the genome. So there are 22 of them here and the X chromosome on the right. And each little circle is a place in the genome where geneticists have found a robust association between what genotype you have at that spot and a different phenotype. So it could be height, it could be weight, it could be diabetes. All the different colors represent sort of different types of phenotype. And in summary, there are now tens of thousands of these places in the genome that have been discovered. Mostly, at least so far, the variants that have been discovered have what are called allele frequencies, so how common they are in the population, above 1 to 5%. And what that means is essentially, if we looked in this room, we would find both versions of the variant. So the versions are called alleles. So we would find both the A and the C in this room. And as I said, the effects of each of these variants are typically quite small, but as I'll try to show you, the impact of the biological discovery may be quite large. And one of the pieces of evidence for that is that where we know, for diseases, where we know the gene that a drug is acting at. So there's a treatment of diabetes, and we know that the gene KCNJ11 encodes a protein, and that's where that drug acts. And what we find is that when we do the genetic association study, and when you do it across the whole genome, it's called a genome-wide association study, or GWAS. When we do that, searching across the whole genome, we actually find variants right at the gene that is the target of the diabetes drug. And that's true for many diseases, and in fact, even for things that we use to treat short or sometimes tall stature. So the hope is that if we do this unbiased search across the entire genome, we'll find not just the biology we know about, but we'll find new biology, new genes that are associated, and those would be new therapeutic leads. And in fact, people have done a systematic review of drugs that were taken into phase 3 clinical trials, so that had gone through a tremendous amount of work, and all that was left to do is figure out, do they work or don't they work? And it turns out that the odds of them working are much higher if there's genetic evidence supporting the gene where that drug acts. So if we could find new genes that are supported by this sort of study, those would be much better drug targets for drug development. So again, even though they have modest effects, that doesn't mean that their biological importance is also modest. I've already told you that these common variants fly under the evolutionary radar, and because they do that, we know that they've flown under the evolutionary radar because they're common. So that means that their effects on biology must be limited. Otherwise, evolution would have noticed them and would have cared about them. But that doesn't mean that if you have a bigger perturbation of that gene or that biology, that it wouldn't have more profound effects. So that's illustrated here. So if you have a variant with a modest effect and it sort of just slightly perturbs a gene, maybe it's expressed a little bit less in the pancreas or something like that, you might have a slight effect on the outcome. So maybe you're 5% less likely to get diabetes. But if you have a variant that perturbs that gene greatly, like it breaks it completely, you don't make the protein at all, you might have a dramatic effect on the outcome. And similarly, you could have a pharmacological intervention which would essentially mimic the effect of essentially breaking that gene or turning it way down, and that also could have a very big effect on the outcome. So you could find a target from the common variant with a small effect, and that might be a very useful target to develop a drug for. And as I said, we know this is a causal path, so you tweak the pathway here by affecting the gene, maybe with a common variant. You see an effect here for the phenotype. But The hard part is we don't know what the steps are in between. And because of that, we should expect that the time frame of translation is actually going to be long. So even where we kind of know something about the biology, so for example, cholesterol, after we knew about cholesterol, it took 4 decades and a couple of Nobel Prizes before there was enough work done to understand that there was a disease gene that affected cholesterol metabolism in a very rare disease called familial hypercholesterolemia. And then there's another couple of decades and another Nobel Prize before a drug was developed, and this was the drug or the class of drugs called statins, which lots and lots of people take now and is sort of a mainstay of therapy to prevent myocardial infarction. But this took many decades. So we think we're doing some things to help speed this up, but again, we need to go from biology from the genetic signals to biology. So we're actually in some ways often even one step behind where we were for understanding cholesterol biology. So this is the way that, on the left here is the way that the results often look is something called a Manhattan plot. You can imagine these are sort of look like skyscrapers here. And on the x-axis of this plot are millions of different places in the genome where there's a variant that's common enough that you would see it in this room. And on the y-axis is how strongly we see evidence of association of that variant with something like height or weight or disease risk. And the challenge is to go from these statistical results saying, oh, this is a really interesting place in the genome, to understanding what the biology is that we should be pursuing. So part of the reason that's difficult is shown on the next couple of slides. So here's a Manhattan plot again, and if you zoom in, what you'll see is that there are a bunch of different variants here, all of which have basically the same evidence of association with, in this case, obesity. And so that already creates some uncertainty as to which the actual variant is. And that can stretch over many different genes. Each of these little things here is a different gene in the genome. And you remember that I told you that most of the variation is non-coding. And it turns out most of the variation that we find as associated is also non-coding. There's a little bit of enrichment for coding variants, but most of the associations are non-coding. And because we don't understand nearly as much about the non-coding portion of the genome, it's actually very difficult currently to connect these variants with which of the genes nearby are they influencing. So that's a big challenge, and it's a huge major initiative that's ongoing right now, is to try to figure out how to make those connections between genetic associations and biology. So this is the challenge, many genes near an association, and if you look at them, there's often few connections to known biology, which is exciting because it means we're finding new biology, but it's also daunting because it means we actually have to start from scratch. So how do we do this? How do we move from genetic associations to human biology? So one approach that we've taken is to try to learn from a trait where at least we have something going for us, and then learn how to do it, and then apply that to other traits and diseases. And so the trait that we've used is actually height. Turns out height has been studied for over a century by geneticists. In fact, even before the word genetics was even coined, people were using height to understand inheritance. And it was used to develop a whole bunch of statistical methods, some of which are illustrated on this slide here. But the reason that we found it to be a particularly good trait to study is that there's a lot of known biology. So in some sense, we had kind of what are called positive controls. We knew that there were some genes that we should be able to come up with. There's about 300 of these that, like the gene for achondroplasia, cause monogenic disorders of skeletal growth. It's highly heritable. I told you that 80% of the variability is due to genetic factors. And there are large samples available because pretty much anybody who shows up to a study has their height measured. And of course, as a pediatric endocrinologist, I was particularly interested in understanding the biology of growth. So how do you go about discovering the genetic variants that influence height? Well, I told you that some people have rare genetic causes of short stature, and they have rare variants with more of an influence, and I'll tell you a little bit about efforts to study that. But most of the variation in height actually comes from, that you see in the general population, or most of the variation in the height in this audience, comes from lots of genes all acting together in a single person. And so because these variants typically end up being common and typically end up having small effects, we knew we needed a lot of people, data from a lot of people to find these variants. And so we formed a consortium called the GIANT Consortium. And these are all the different studies that we're participating in. Each of these studies has multiple different investigators working on them, so it's hundreds and hundreds of investigators, and it's been a lot of fun working in this international consortium and having everybody work together towards a common goal. So putting all that data together, each of the studies sent us files, and we had to go back and say, oh, this doesn't quite look right, can you fix it? And the group that was doing that is shown here, and for height, the leaders of that effort are Tonu, Andy, and Sailaja. And the results from that effort, it's now actually a total of about 700,000 people, but here are some actually slightly earlier results. The Manhattan plot are shown here. And in summary, with that studies of 700,000 people, we've now discovered about 900 different places in the gene, 900 different variants in the genome, and they're actually in about 400 different places. So there are a lot of places that have many different variants. So they're saying, right here, right here, right here is important. You should go study this place. We took a special effort to try to find variants that change the protein coding sequences because of this problem of going from non-coding variants to genes. And we found about 100 of them. But even despite those efforts, most of what we found was non-coding variant. And together, if you add up all of these variants together, even though each one of them has a very small effect on height, together we can now account for about a quarter of the variability in height. So about a quarter of the variation that you see in this room, we could predict that, about a quarter of it, based on just the genetics that we already know about. But the main goal is really not so much prediction, but to try to understand the biology. So here's one example. There's a group of genes that act together in sort of a common biological, what's called pathway. The end of that pathway is signaling by something called IGF, or insulin-like growth factor. This is how growth hormone works. So if you get growth hormone, your body makes more IGF. And if IGF acts in the bone, your bones grow more. So IGF actually doesn't sit around usually in the blood just by itself. It's actually usually wrapped up in these binding proteins, in this case IGF-BP4. The IGF binding proteins themselves are broken down by another protein called PAP-A. And that protein, PAP-A, is itself inhibited by another protein called STC2. And if you go, the inhibited of this blocks that, breaks down this, and you sort of trace it all back, what you predict is that if you have less STC2, you should have more of this IGF-1 that's free to act and act on the bone, and you should get more growth. And in fact, what we found were there were two rare coding variants in STC2, and our colleagues were able to show that STC2 was, with those rare coding variants, was less effective at inhibiting PAP-A. And in fact, the people who had these rare coding variants were taller, in this case by about 1 centimeter or 2. So that validates that pathway. So there was a case where we were lucky. We had a couple of rare coding variants both pointing to the same gene. We were wondering, could we use the non-coding variants to try to understand biology? And there are computational methods where we can look across the genes that are in different places in the genome. And so if you make a plot using something called GRAIL that was developed here at the Broad by Shomer Rashuderi and Mark Daly's lab, what that does is it actually mines the published literature. So instead of me having to read all 100 million articles that have ever been published, which I'm working on, basically GRAIL has done that for me, and it looks for commonalities between articles and that mention each of these genes, and where it can find similarities between them, it draws a connection between the genes. And what you can see is that lots of the genes that are associated with height, or that are near variants that are associated with height, actually cluster together. And when you look at which genes are clustered together, they actually make sense. It's recognizable biology. So this is the growth hormone IGF pathway that I just showed you. And then there are other pathways, like here's one, TGF-beta signaling. It turns out that that pathway is disrupted in a disease where you grow too much, called Marfan syndrome. So then we did it with our next round, where we had even more variants. And what you can see, there was even denser spider web of connection. And a lot of the pathways that we hadn't recognized when we had fewer places in the genome, fewer variants, now all of a sudden emerged from these connections. And again, these were relevant pathways that we know are important for growth. So essentially, the more variants you have, the more commonalities you can find between the genes near those variants, and the more biological understanding you can get. But that being said, even though we have this dense spider web and we are identifying all these interesting known pathways, many of the variants still have no known connections to biology of human growth. So that means that even though we understood a lot about growth from these monogenic disorders, there's actually a lot we still need to learn, and that we can learn by studying the genes that are near these variants. And one of the ways we're trying to do that is by developing new computational methods, and There's a method, this is developed by originally Tuna Pears and his colleagues, called DEPICT, and I'll tell you a bit more about it later. And then this is work by Rebecca Fine to adapt it. And essentially what's shown here is a different way that we can look at our results. This little what's called a heat map here. The rows are all the different genes that have variants in them that affect, I'm sorry, the columns are different genes that have variants in them that affect height. And the rows are different kind of sets of genes that keep showing up over and over again when we look across the different places in the genome that are associated with height. And the color here is how strongly each gene is predicted to be a member of each of these sets of genes. And so what you're looking for are these little blotches of red where there are lots of genes that are all strongly involved in the biology that's been implicated. And so this little patch of red here, if you zoom in on it, if you pay attention to these little black and gray bars here, the black ones are genes that are already known to be important because they have mutations that cause severe syndromes of disordered skeletal growth. But then there's these other genes in red boxes here that actually aren't known to be important for growth and that we strongly predict are in fact new growth biology genes. And that prediction was in fact borne out after we made these predictions. There was a paper that was published that one of these genes actually causes a severe growth disorder. So this computational method seems to be doing a pretty good job at finding new genes that we could study. And these methods are completely applicable to everything else, not just height, but disease risk, obesity, everything, and we and others are doing that. So I mentioned that some children have rare causes of short stature. So we also have ways of trying to find these very rare variants, so things that even if we looked in everybody's genome in this audience and 10 more auditoriums like it, we wouldn't find it. So for that, what you have to do is you have to start with the folks who have short stature and look through their entire genome and see what variants you find. And so Andrew Dauber, in collaboration with lots of other people in the lab, recruited such patients with short stature, both here and in Cincinnati, where he moved to, and then looked through the data and then analyzed that data actually in collaboration with Eliana and others to try to find the causes of short stature. And we were actually much more successful than we thought we were going to be. So it turns out that if you take people with severe short stature, so 6 inches below average or more, about a third of patients will have a single rare variant that basically explains why they're short. And I'm not going to go through all the different things that we found, but at least some of them actually had some potential therapeutic implications where if the children had been diagnosed earlier, there was a syndrome of short stature and intellectual disability that is possible, at least in theory, that they could have had some treatment for it. So I mentioned briefly prediction for height. So I said that we could explain about a quarter of the variation in height in the population. So can we use that in any way? Well, the short answer is, first of all, not yet. This has not come into the clinic. But I'll give you an indication about a way that it might in the not too distant future. So this is an experiment we did about 6 years ago. It was led by Rigel Chen, who was a grad student in the lab. And what we did was we took people who were very short or very tall. And at the time, we took all the variants that we knew about at the time, and we determined the genotype at each of these variants for all the tall people and all the short people. And then we said, well, we know how much these variants are supposed to influence height. So how many sort of taller versions of genes should we find in the tall people, and how many shorter versions of genes should we find in the short people? And does that match what we would expect? And for the tall people, it matched perfectly. But for the short people, their genes weren't as short as they should have been. Or to put it another way, they were shorter than their genes predicted. And so what that suggested to us was that there are some people who are discordant, where if you look at their genetics that we've discovered, they're much shorter than they should be. And what we think is that those folks have something else going on, either a big effect variant or maybe there's an underlying disease that we don't know about, like celiac disease or something like that, that's influencing their growth. And so if we could identify the people who are short but their genetics say they shouldn't be, those might be people that we should do more clinical work on. Okay, I'm going to tell you one more quick story about height and then a bit about obesity and then finish up. So this is from not one of my favorite movies, actually. Sorry, Rocky franchise fans. But this is Sylvester Stallone on the left, who is of Italian descent, and then Dolph Lundgren, on the right, who's playing a Russian, but I think is actually Scandinavian. And they obviously have a big height difference, but in fact, folks with Southern European ancestry are on average a little bit shorter than folks with Northern European ancestry. And it turns out that we were able to use the genetics that we had discovered from height to show that part of the reason for that is that of these many variants that influence height, a lot of them systematically, the versions that make you taller, became systematically a little more common in Northern Europe than in Southern Europe. All right, so I'm just going to finish up with a bit of obesity. There are lots of different measures you could use to study obesity, and we studied each of these to some degree, but I'm going to tell you about the work on the simplest of those, which is body mass index. It's your height, it's your weight divided by your height squared, essentially. And we did the same sort of work that we did for height, genome-wide association studies. Here's a Manhattan plot. And when we looked, there were few obviously recognizable genes. And even when we used the computational methods that were around at the time, they didn't give us any hints as to what the biology was except for a few that were in a pathway, in a set of genes that we knew about that affect the nervous system and probably the brain and probably influence either appetite or energy utilization. But most of the variants had no known connections to body weight regulation. And so we developed a new computational method, which we called DEPICT. This was done by Tuna Pears in the lab in collaboration with folks from Groningen. And this had as a goal to figure out which the genes were near associated variants and to identify sets of genes that together were affecting obesity and also where those genes were acting, like what tissues were they acting. It was the brain, the gut, the fat cells. And so we did that, and if you try to look at what tissues it is, it turns out the genetics is very firmly pointing us, at least for body mass index, to the nervous system. We think that's the brain, although actually in the intestines have their own nervous system, so it could be that also. And then when we try to find sets of genes that are important for weight regulation, we got a whole bunch of different sets of genes, and when we zoomed in on one of them here, and there are lots of sets of related genes here, we were actually quite interested in this because these genes pointed to a particular aspect of biology in the nervous system, particularly having to do with something called glutamate signaling. It's an amino acid that your brain uses to have neurons talk to each other. And there's a drug called topiramate, which was not too long ago approved for treatment for obesity, that actually nobody has a clue why it works. Why in some people, they actually get effectively anorexia and they lose weight. It was originally designed for epilepsy and was discovered kind of coincidentally to cause weight loss. And one of the proposed mechanisms of topiramate is actually right at this set of genes. So we think that this may be how topiramate might be acting, and we're looking into that. And the idea is that if we could discover exactly how topiramate works, it has a bunch of other side effects that people don't like. Maybe we could design something that just affects the weight loss. So moving forward, we're doing more genome-wide association studies. We're getting to variants that are a little bit rarer, so maybe only once in the population, doing those with about 3 million people. And just as a sort of preview, when we've put about 1 million or a little less than 1 million of those people together, we now have 3,000 different variants that affect height, 700 that affect body mass index, and we have a big increase, particularly for BMI and the amount of heritability that we can explain. So this is a lot more potential biology to explore. So in summary, I've told you we've been using this approach, largely genome-wide association studies. We found many, many genes that influence our height and our body weight. Most of the variants we found are not coding variants, and they have small effect sizes, but that doesn't mean that the genes near them aren't important. We think they are. And as we get more and more of these associations, we're getting a clearer and sharper picture of the biology, which we think will now provide new therapeutic leads that are more likely to lead to successful treatments. And again, this approach is not just for height and obesity, but also for many other diseases, some of which have been talked about in this lecture series. It's a hard road to go and a slow one to infer the biology, but it's an important one, and many of us are working on this. And we know that we're on a right track because the genetic association ensures a causality. So I've tried to acknowledge some people on the way. These are folks who either are or were in the lab, and many of them are faculty elsewhere now. A lot of the work I talked about is from the GIANT Consortium, which has been great, and these are just a few of the leaders of the GIANT Consortium and then some of our collaborators here. So thank you very much for your attention. I'd be happy to take questions.
Thank you very much, Joel. If anybody has any questions in the audience, please come down. We have 2 microphones here in the wings. Come on down and ask. And as a reminder to our Facebook Live audience, if you have any questions, please type them in as comments into the feed, and I will get to them as best I can.
Thank you very much for your wonderful talk. So I'm interested in study of genetics of longevity. So my question is that would you please give some comments on the transferring your methodology to a study of longevity and some potential pitfalls, the main difference, the bottleneck, and some hyperfall suggestions. Thank you.
Yes, a great question. So as I said, this is a general methodology, right? You know, the approach is exactly the same. How successful it is depends on a couple of things. One is just what the genetics turn out to be. So for those of you who are paying attention, the sample sizes that we used for height and for body mass index were about the same, but we were much more successful for height. That's partially because height is more genetic, but it's also partially due to just the underlying what we call genetic architecture. So longevity, it's been tried, and I should say the other thing is what sample size you can get. So we're in a great position for height and body mass index. It's a little harder for longevity, especially if you're looking for people at the extremes, so like centenarians, who people who've lived to over 100. But that being said, people have done genome-wide association studies, and they found things. The biggest signal that they found for longevity is in a gene called ApoE. So the variant ApoE4 has lots of different effects. The most sort of well-known is that it increases your risk of Alzheimer's disease, but it also increases your risk of cardiovascular disease, and that's the biggest signal that you find in longevity. The difficulty with longevity is that there are lots of different ways that you can not have longevity. Any of those will show up. So, you know, all of the things that affect diabetes probably also affect longevity. All of the things that affect Alzheimer's disease also affect longevity. So it's kind of a common pathway, and so you need things that have big effects on big killers to see them. That being said, it's perfectly possible to do, and there are clever ways of getting around the fact that you have to, you know, the ideal thing would be to survey a whole bunch of people and then know how long they lived. That's tricky to do for obvious reasons, but you can do tricks like how long did their parents live, and you can sort of infer like how long they might eventually live. So it's being done. It's just hard.
Thank you.
I have one question about the causal variants, because when you're looking for causal variants in the DNA, do you only use GWAS, or you combine it with other types of data like eQTL?
Yeah, so when we're trying to, so let me unpack that question to a couple things. So there's, first of all, just is there causality at all? And that we know that something in the region is the causal variant. And if you've used GWAS or sequencing or whatever, you know that there's something there to look at. But then there's try to figure out what the actual mechanism is, so what the root of causality is. And that's where things like what you called eQTLs are. So what those are those are places in the genome where there's a variant, and not just affects, say, your height, but you can also figure out that it affects expression of, you know, of the 10 genes nearby, it only affects expression of this one. And so that would say, okay, that's probably the right gene. There's all sorts of complexities to combining eQTL data and genetic data in ways that can fool you, but that is one approach that you can take, absolutely. And there are lots of other data sets that you can try to pull in to understand mechanism.
Thank you for the very interesting talk. I have a question about obesity. In recent decades, it's been increasing so that about 40% of the adult population in the United States have obesity, and some subpopulations even more prevalent. But the genes presumably haven't changed. What is causing the epidemic?
Yeah, so that's a great question. You know, there's sort of a simple answer to that, which is, you know, more food and less physical activity, or maybe, you know, there's a more complex answer, perhaps, of different types of food. I would say the short answer is we don't really have a full explanation of that. The one thing we do know is exactly what you said. It's not a change in the genes that we have, right? The change has been over way too rapid a time scale. So even though evolution can act rapidly, not that rapidly. And so what's actually interesting is when people look at what fraction of the variation in body mass index is explained by genes, it's actually going up. And that's because our environment has been getting more and more homogeneous. Essentially, it's been getting more and more what's called obesogenic environment, obesity-causing environment, and that's just becoming uniform now. So there's less environmental variability than there used to be. But what's changed exactly? Lots and lots of things have changed. We're much more sedentary. We eat completely differently than we did 30 years ago. There's, our microbiome is different. That's a super interesting problem. The problem is we don't even know what to measure often. So one potential route that genetics could help is if we understood the biology, we could then understand what environmental factors that biology is likely to be interacting with. Or we could look for what's called gene-environment interactions. So are there different things, or different types of foods that people eat that only make you heavier if you have certain genetic patterns? But we're very much in the infancy of trying to answer your question.
Thank you.
I have a question. I think it concerns the question of causality that you talked about. I wonder, when you look for genes in association studies, you find genes that have a very tiny effect of less than 1% of the natural variation. What makes you hope that a drug that targets this gene could possibly have a large effect, which is what you actually want?
Yeah, so thank you for the question, and this is a common concern that's raised about genome-wide association studies. And you're right, actually, in fact, 1% of the variation, we would be jumping over the moon if we found something that big that explained 1% of the variation. So the reason that we're hopeful is just the examples that already exist. So I mentioned the statins. They act at a gene called HMG-CoA reductase, or a protein called HMG-CoA reductase, which is encoded by the HMG-CR gene. Turns out that there's a common variant in the HMG-CR gene which affects a tiny fraction of a percent of your LDL cholesterol, of the variation in LDL cholesterol. But statins actually do much more to that gene than the common variant does. And again, the common variant has that small effect because probably having a big effect on a major gene for cholesterol synthesis is not something evolution would like very much. And so those variants are just super rare and may not even exist in people. So the only way of finding that gene genetically is through these common variants of small effects. But then when you develop the drug against it, you can break it more severely with a drug. And particularly there's some things which they might be very important during early childhood development or maybe even during embryogenesis. And so there's no strong genetic variation, but you can actually give the drug in adulthood and it turns out to be relatively safe. You just wouldn't want to give it to, say, a developing fetus.
Yes, my question goes in the same direction of when you want to investigate the therapeutic approaches. You said that, commonly we investigate the genes that are implicated in the same pathway, right? But how can we know if the other genes that weren't previously reported to be implicated in common pathways, how can we know that they might not influence the therapeutic approaches?
So let me make sure I got your question right. Are you asking how do we know the genes that we know nothing about? How do we know if they're in the same pathway? Yeah.
And their implication on the...
And how do we know? Right. So I'll answer both of those. So how do we know it's in the same pathway? Actually, that's why we developed DEPICT. So if you look, most of the genes in the genome, nothing is known about them. So there's a huge amount of biological research, and it's focused on a very small subset of the genes in the genome. And that's because somebody discovered using, say, genetics or a different approach, this is a really important gene, and so everybody knew, oh, that's a really good area to work on. So tons and tons of labs all work on that same protein. And there could be 6 genes next door that nobody knows anything about, and they have names that don't mean anything to anybody, and nobody works on them. So what we did is we actually looked for commonalities in which tissues and which cells and which conditions genes were turned on or off. And then we said, which genes look like the genes in this pathway? And so we were able to annotate pathways, basically every gene in the genome, give it a probability for is it in this pathway or not. So that's one approach, and people have taken other approaches. But then do other genes in that pathway, are they good therapeutic targets? It's tough to know until you try, but at least you know that biology is likely to be the right biology. There certainly are examples of drugs that hit multiple sites in a pathway that are all effective, but sometimes not, and that could be in some cases we actually don't understand enough about the biology to know which of the members of that pathway we should be targeting and which ones we shouldn't. One clue sometimes can come if you have a variant that's right in that gene, that probably is a little bit more evidence that that is the gene to target in the pathway. You can also look at the effects on other phenotypes. So if you have something that, for example, lowers lipid levels but raises blood pressure and raises your risk of diabetes, maybe that's not the gene to go after. So that's another way that you can try to get at it.
Sorry, another question.
Yeah.
Is there any epigenetic implications?
So role of epigenetics. So epigenetics, there's sort of two classes of that. So what epigenetics is so there's changes in your genetics. So you, instead of having an A, you have a T at some place. But then there are other things that can happen to the DNA that don't change the sequence, but have like other chemical groups stuck onto it, like a methyl group, or they might have other proteins that are stuck onto it. And those are what are called epigenetic changes, so sort of around the genes. Most of those are not inherited. So essentially your epigenetics gets erased when you're at fertilization, and then it gets rebuilt from scratch. So most of the epigenetics won't show up in the methods that we're looking at here. There's some hint, more in mice and plants, but some hints in humans that a few things can be inherited for at least a few generations. Those are also difficult. We're racking our brains still on how to get at those and trying to use like multi-generational studies, and nobody's made a lot of headway, I would say, in humans on it. But what can happen is that the epigenetics can be the mechanism by which the genetic variation acts. So it might be that you have an A or a T here, and that influences whether some protein now sits down on that part of the genome or not and turns on some gene next door. And you might see evidence of that in epigenetic changes that go along with the genetic change. So those were called, if you're looking at methylation, methyl QTLs or epigenetic QTLs. And that's one of the methods that's sort of one of the other data sets that we'd like to bring in to try to give us clues as to the mechanism. So I'd say it's more sort of what's downstream, and genetics affect epigenetics, and environmental influences can affect epigenetics. And so this can be sort of one of the clues of the downstream mechanisms.
All right, well, I'm sure Joel will be happy to stick around for a little while after the talk to take any other questions that you might have. Thank you, everybody, for joining us tonight, both here in the auditorium at the Broad Institute and on Facebook Live. Please join me in thanking Joel for tonight's talk and the Hektor family.
Great. Thank you very much.